comes science. They might be trying to here comes science.
Hi, just wanted to jump in here at the beginning because there was an issue with the editing of the video. For most of the video, my face is missing. Something happened with the exporting. I do believe I show back up around 20 minutes or so in, so at the very end, you will see me again. So see you in the next video. Hi, Dr. H here. This will be our Unit 4 review, and this will cover cell communication and cell cycle. So we'll jump right in, and the first thing we'll talk about will be cell communication. In general, when we are talking about cell communication, the first thing we want to look at is how far apart are these two cells that are communicating. This distance can really be anywhere from cells being very, very close together, as we see here with our paracrine signaling, which is cells in the immediate area picking up a signal, to cells that are, one-on-one very close, that we see with our nervous system and the synaptic signaling. And we can also see communication between cells that are very far apart in extreme different areas of the body, and that would be our endocrine system, our hormonal system. So we have very, very close signaling, even to the extent that cells would actually physically be making contact with each other to communicate, all the way up to cells on complete opposite ends of the body being able to communicate. When we look at cell signaling, we have three basic steps that brings this information from where it is received to where the cell is eventually going to respond. Step one is reception. This is the cell picking up this signal, whether it is some sort of chemical signal as shown here, or remember, cells can also respond to other types of stimuli, such as sound, or light, or temperature. Then in the second stage of transduction, this message is relayed from the site of reception to where the actual third stage will occur, and this is the cell's response. Okay, so we're going to talk a little bit about reception and some different receptors. We'll talk a little bit about signal transduction pathways. We're not really going to get into response all that much, since response is highly cell dependent. So starting off with receptors, receptors can be found either on the surface of the cell or inside the cell. And this is going to depend on the type of signal that these receptors are picking up. If it is a chemical signal, if that chemical signaling molecule, that ligand, cannot enter into the cell easily, cannot diffuse right through the membrane passively, then the receptor will be on the cell surface. If the signaling molecule is able to move through, so if it is a non-polar molecule, if it is a small molecule, then we will often find the receptors to these types of molecules inside of the cell. A few examples here very quick of some cell surface receptors. We have these G protein coupled receptors. They are referred to as G proteins because the energy used to start the signaling process comes from GTP rather than ATP. We also can see ion channel receptors, which when they are stimulated will open and allow ions to pass through the membrane. And our final type of cell surface receptor is our receptor tyrosine kinases. And these receptors have this enzymatic activity, this kinase activity, that is only turned on, only activated when the signaling molecule is bound. Okay, so all of these types of receptors, those three basic types, are all found on the cell surface. As a quick example of a receptor that would be located inside the cell, Here we have an example of the testosterone receptor. Okay, testosterone is a nonpolar molecule, so it is able to diffuse easily into the cell, so the receptor is found in the cytoplasm. One other thing to remember about receptors is they are often targets for drug action. And these drugs will either activate the receptor or they will block the receptor. And we refer to these drugs as agonists or antagonists. Okay, an agonist drug is going to mimic the natural ligand, the natural signaling molecule of this receptor, so it's going to increase the activity, increase the signal from that receptor in the cell. The antagonist type drug is going to block the activity, so it's going to shut down the cell's response to this signaling molecule. So many drugs work here on these receptors as either agonists or antagonists. Okay, one more very important aspect of receptors is to remember that since these are proteins, they have the potential to have a mutation. Okay, remember, a change in the genetic sequence could potentially lead to a change in the receptor function. So these mutations can either be activating or inactivating, and that will either lead to an increased activity in the cell or a decreased activity. And there are many diseases that are caused by these receptor mutations. So that's step one, our cell signaling, reception of the signal. Step two is our signal transduction pathway, right? Bringing that information, bringing that message from the receptor where it is initially picked up to where the cell is going to respond. And these signal transduction pathways are multi-step, okay, and they often involve this phosphorylation cascade, where each of these steps along the way is a kinase enzyme. Right? A kinase enzyme is going to transfer a phosphate group from ATP to a protein and phosphorylate that protein. And this phosphorylation will then activate the next kinase in line to phosphorylate the next target. And we can have here, we see a couple step pathway here. They can actually be much longer than this, 5, 6, 7 steps even. And this multi-step pathway is very important for the cell, mainly to amplify the signal. Remember that all of these steps here are enzymes, and enzymes can catalyze multiple reactions. They work over and over and over again. So if we imagine that each of these steps can activate 10 or 100 downstream targets, then each step is going to make the signal 10 or 100 times stronger. So what is a very weak signal, a very low concentration of this signaling molecule outside of the cell, can actually lead to a very strong, a very robust cellular response because of all of these amplification steps. Another very important aspect of signal transduction is the action of second messengers. Okay, and these second messengers are small molecules that can rapidly diffuse through the cell and help to speed the message along. So here we see an example of a transduction pathway that is actually using two different second messengers. First up at the cell membrane from the initial signal, we have this molecule called IP3 that is produced, and that moves and stimulates the release of calcium. And calcium is another very common second messenger molecule. Okay, so remember, these second messenger molecules are small molecules found inside of the cell that can rapidly diffuse and help to speed this message along. Okay, one additional very common second messenger is cyclic AMP. Okay, and here we see this being formed. Again, there's a couple enzymes here, and this cyclic AMP can rapidly move through the cell and helps to speed this message along. Okay, so that is step 2 of the pathway, signal transduction. Like I said, I'm not going to get too much into response here, just because it is so cell specific. But I do want to talk very briefly about a few different examples of communication at these different scales. Right? Remember, going from cells very, very close to each other, maybe even in contact with each other communicating, all the way up to cells in different parts of the body communicating through the use of hormones and the endocrine system. So our example of cells being very, very close to each other happens in the immune system, and this is contact-dependent inhibition that we see with our cytotoxic T cells, where an infected cell will display a little piece of the pathogen on the cell surface, and the cytotoxic T cell has receptors that will come along and physically bind to that and recognize that this is an antigen, and will then kill this infected cell and thereby help to stop the infection. Cells that are a little bit further apart, still very close but not actually touching. This is an example of local communication that we would see in the nervous system with our neurons and the synapse, which is where the two cells come together. Okay, so these two cells are very, very close to each other, but they are not actually touching each other. So when the signal gets to the end of the neuron, the chemicals are released, the neurotransmitters are released, and they bind to the receptor on the next cell in line, and that cell then picks up the message and responds accordingly. We spent a little bit of time in class going over the action potential in neurons, so I think this is probably a good idea to look this back over. Remember, it is all due to the membrane potential, with the sodium ions being a high concentration outside the cell and the potassium ions being a higher concentration inside of the cell. This is due to the action of the sodium-potassium pump. Right? This pump moves 2 sodium ions out of the cell and brings 2 potassium ions into the cell. So since there's this uneven exchange of the positive ions, the outside comes positive, the inside is relatively negative, and the neurons are able to manipulate this membrane potential by opening and closing these sodium or potassium channels. The action potential is begun through the opening of sodium channels, and this allows sodium to come rushing into the cell. Remember, sodium is high outside, so it's going to move down the gradient through these open channels, and that causes our rapid depolarization of the neuron. Then, to get back to the resting potential, the potassium channels are opened, and the potassium goes rushing out of the cell, because potassium, remember, was at a higher concentration inside, so it will move down its gradient and move out of the cell. That is our local communication, right? Cells that are very close to each other, able to communicate. And our last example of communication is our long-distance communication, and this is the hormone system, the endocrine system in our bodies. Okay, hormones are released from the endocrine glands, which are located all over the body. The hormones, the signaling molecules, enter into the bloodstream and travel throughout the entire body. And cells that have the receptor for these hormones can pick up that signal, and then they will respond. Other cells that do not have the receptor will not respond to the hormone. Right? The cells need to have the receptor in order to pick up the signal and respond. These hormones work in these feedback loops, and they will often work in pairs. Right? The example, kind of the general example that we use a lot to talk about these feedback loops is heating and cooling a house. Okay, if we start at a certain set point where we want the temperature of the house to be, if the temperature falls below that, then the thermostat will turn the heater on, the temperature goes up, and that turns the heater back off. If the temperature gets too hot, then it'll turn the air conditioning on, bring the temperature down, and that will turn the air conditioning back off. Right? So these are examples of these negative feedback loops where the end product turns the system off. Okay, so, and a real quick example of this, this is the insulin glucagon system that responds to the amount of sugar in the blood. If sugar is raised, like right after a meal, then the body produces insulin. Insulin signals the cells to take that glucose out of the blood, bring it into the cells, and use it for energy. If the glucose levels fall, such as during exercise or fasting, then the body releases the enzyme glucagon, which causes the body to break down some of its carbohydrate stores and release glucose monomers back into the blood. Okay, so both of these hormones are working together to bring the body back to this homeostatic set point. Okay, hormones will often work in pairs like this to maintain this homeostasis. That is the first part of our unit. That was cell communication. The second half of our unit, we got into the cell cycle and cell division. Remember, cells divide for three basic reasons. For unicellular organisms, this is how they reproduce. Growth and development, as multicellular organisms grow larger, they will add more cells to the body, and if those multicellular organisms are damaged, if you have tissue damage, then the cells will divide to replace those damaged tissues. Real quick, before we start talking about cell division, we need to talk about a few of these terms that are very easily confused, that are used in describing the DNA structure and the DNA packing. DNA exists in what is called a loose state for most of a cell's life, and this structure is what we call chromatin. Okay, and in the diagram here, this would be our 30 nanometer fiber. So there is a decent bit of structure here, but it is considered loose DNA. Then as the cell begins to move through the process of division, this chromatin will begin to condense and form our final chromosome structure that we only really see during cell division. Okay, and these chromosomes, since the DNA has been replicated before the cells divide, they are what we call replicated or duplicated chromosomes. And they are made up of two separate linear pieces of DNA that are referred to as sister chromatids. It's very important that you are able to keep all of these terms straight, like chromatin, chromosome, chromatid. Right? Make sure that you are using these terms correctly. The cell cycle here is our basic overall pattern of cell growth and division. It's very roughly broken into interphase, which the cell will spend most of its life in, and then the mitotic phase, where the cell is actually dividing. Okay? And then interphase is broken down into three separate stages, G1, S, and G2. Okay, in G1, the cell is growing and preparing to replicate the DNA. During S phase, the cell is actually replicating the DNA. G2 is a short little rest phase, making sure that everything is ready for the cell to actually divide. So the cell will check over the DNA, make sure it is all copied correctly, and then the cell will enter into mitosis, where the cell will actually go through the division process. Okay, so we're going to talk mainly about the mitotic phase here. There are a lot of key events here that are happening in the nucleus as the cell divides. I don't really think you're going to need to memorize these stages and exactly what happens in each of them, but it is a good idea to get a rough idea of what the DNA is doing, what some of the other cellular structures are doing as the cells move through this mitotic process. As the cell moves through, DNA will condense from the loose chromatin that we see in interphase to the densely packed chromosomes. The nuclear envelope will begin to break down, and we'll see these microtubules called spindle fibers will start to grow from either end of the cell. Okay, and these spindle fibers will go in to the chromosome area, and they will attach to a chromosome. One spindle fiber from each end of the cell will attach to each chromosome, and these spindle fibers are used to move the chromosomes around. First, they will get all the chromosomes lined up along the center of the cell, and that is very important in cell cycle progression. Once all of the chromosomes are all lined up, the spindle fibers will begin to shorten, and that will pull the sister chromatids apart. So now we no longer have these duplicated chromosomes, and once these chromatids are separated, the cell will go back to its interphase state. So the DNA will begin to decondense, the nuclear envelope will reappear, the spindle fibers will break back down, and we'll be back into our G1 stage. Our very last step of mitosis is cytokinesis, and this is the actual splitting of the cytoplasm into our two daughter cells. In animal cells, this is accomplished by having a ring of protein grow around the cell and squeeze it into and pinch it into two separate cells. In plant cells, since they have the rigid cell wall, they cannot just pinch in half. So there is a new cell wall laid down between the new nuclei. Okay, the final topic is, how is the cell cycle regulated? Okay, the cell division needs to happen only when it is needed, and it needs to stop at the correct time. So there are these checkpoints as the cell goes through the cycle. And if the cell successfully meets these checkpoints, then the cell can move on to the next stage. If the cell does not meet the requirements of the checkpoint, then the cell cycle will stop, and the cell can either fix the problem, or the cell, if it's a major problem, then the cell can actually eliminate itself through apoptosis. So the mechanism for these checkpoints is cyclin CDK complexes. Okay, cyclins are proteins that go up and down in levels as the cell moves through the cell cycle. And the CDKs is a cyclin-dependent kinase. So this is an enzyme, it's a kinase, that is only active when it has its cyclin partner bound to it. So these cyclins, as I said, they go up and down through different phases of the cell cycle. We see a few examples here. And as these cyclins complex with their CDKs, the CDK enzyme activity, the kinase activity becomes activated, and they will phosphorylate their targets, and that will move the cell through that stage of the cell cycle. So here we have our sort of link between the two parts of this unit, our cell communication, our growth factor coming in, telling the cell that it is time to divide. The receptor picks up that signal. The transduction pathway, which is not shown in this diagram, then leads to the production of the cyclin protein, binds to its CDK partner. The CDK is now active, and that active CDK can then push the cell through the cell cycle. It is very important for this cell cycle regulatory system to work correctly. If it is not working correctly, if cells are dividing out of control, then that can progress into a tumor and can then lead to a cancerous growth. So most cancers are caused by an accumulation of mutations in these cell cycle regulatory pathways. So that is it for our Unit 4. Okay, we covered cell communication. Remember the 3 basic steps: reception, transduction, and then response. And then we got into the cell cycle. Remember interphase, mitosis, the basic events of mitosis, and cell cycle regulation. So I hope that all made sense, and I will see you guys again soon.
Science like Galileo dropped the orange.